Well, hello, my fellow pre-meds. In this video, we're going to be discussing the structure and function of enzymes. So I purposefully left out enzyme kinetics from this video because I do recognize that that is a bit of a difficult to digest topic. So I thought I'd divide the discussion on enzymes into two separate videos, although I'd highly encourage that you watch that video because it's extremely high yield and I explain it in a pretty fun to learn way using monkeys and peanuts. But enough with the monkey business. Let's get started with our discussion on the structure and function of enzymes. Did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide deck. Links in the description down below, as well as the links for everything else. All right, so these are some key properties. I'm going to briefly mention them here, and then we're going to dive deeper into them in subsequent slides. So enzymes are biological catalysts. That is to say that they will speed up or increase the reaction rate of chemical reactions that occur in the body. They do this by stabilizing the transition state or lowering the energy at which you achieve the activation energy. Now, notably, they don't impact the thermodynamic side of things, so they don't increase or decrease Gibbs free energy or change in G. All right, so our next key property here is that enzymes are not consumed or permanently altered during the reaction. So while there may be some transient changes to the structure of the active site within an enzyme during the reaction, notably, once the reaction goes to completion, that enzyme will revert back to its natural three-dimensional shape that can participate in another reaction. So you don't have to recycle the enzyme for every reaction. All right, next point here. Most enzymes exhibit high specificity for a particular substrate or group of substrates. So a commonly given analogy over here is that imagine you were in a parking lot, a vast parking lot. Let's say you were in Disney World and you wanted to unlock your car specifically with your key fob. Well, your key fob has high specificity to your car in that it will only unlock your car and no other car. Now, that's a bit analogous to how enzymes work, although there is a bit of difference there because sometimes enzymes can accommodate multiple molecules that are structurally related. All right, finally, enzyme activity is influenced by environmental, pH, and temperature. So as we'll see, there's like optimal pH values and temperatures for the activity of enzymes. All right, before we dive deeper into those key properties, let's talk a bit about classification and nomenclature of enzymes. So these are the broad classifications, or the families, if you will, of enzymes. We're going to discuss them further in the subsequent slide. So oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So the way the nomenclature works for enzymes, most enzymes will end with the suffix ase. Any word that you see on the MCAT with the suffix ase is likely an enzyme. There are a few exceptions where enzymes don't end with the suffix ase. So for example, trypsin or chymotrypsin or pepsin. And then notably, many enzymes will be preceded by their substrate name. So for example, urease acts on urea, or the description of the action they perform. So for example, lactate dehydrogenase will dehydrogenate lactate. All right, so here are the families of enzymes with examples. So oxidoreductases, as the name implies, catalyze oxidation reduction reactions. An example, lactate dehydrogenase. So you can see that over here we are oxidizing lactate, so that hydroxyl group will gain another bond with oxygen. So when you do that, so in Gen Chem, we typically think of oxidation reduction in terms of loss or gain of electrons respectively, whereas in biochem and orgo, you can also think of it in terms of gaining or losing hydrogen atoms or gaining or losing bonds with oxygen. So here we oxidized lactate, so there must be an oxidizing agent, which is NAD+, which itself was reduced, and you can see that by the addition of the hydrogen atom over here. All right, next we have transferases. So transferases catalyze the transfer of one group from one molecule to another. Now, a common example on the MCAT is a group of enzymes called kinases. You can see that they'll transfer a phosphate group, typically from ATP, to another substrate, so they will phosphorylate that substrate. Next, we have hydrolases. So these use water to break down a molecule into two separate molecules. So as you can see right here, urease will break urea into ammonia and carbon dioxide. Next, we have lyases. Again, like hydrolases, these will break one molecule into two separate molecules. However, they don't rely on water. They don't need water. So one example here is pyruvate decarboxylase, which will decarboxylate pyruvate into acetaldehyde and CO2. Next, we have isomerases. So isomerases rearrange bonds within a molecule or rearrange the atoms within a molecule. So they deal with isomers, as the name implies again. So you have stereoisomers and constitutional isomers. Stereoisomers are just like the R and S absolute configurations. And then constitutional isomers are molecules that share the same chemical formula, however, are arranged differently. And an example right here is glucose 6-phosphate isomerase. So glucose 6-phosphate and fructose 6-phosphate are constitutional isomers. So you can see that glucose is a 6-membered ring, whereas fructose is a 5-membered ring. However, if you look at the chemical formula for both of them, it's C6H12O6, but it's just that the atoms are arranged differently. And this is, by the way, one of the early steps of glycolysis. All right, finally we have ligases. So ligases will join two molecules together and they harness the energy from ATP. So in order to join, for example, pyruvate and bicarb, or sometimes you'll see it as carbon dioxide, pyruvate carboxylase harnesses the energy from ATP to form oxaloacetate. All right, now back to our key properties, and we're going to dive a bit deeper as I promised. So recall I mentioned that enzymes are biological catalysts. They stabilize the transition state and lower the activation energy, and notably they don't impact change in G. So there's a lot on the slide, but let me walk you through it. Now first I'd like to draw your attention to this diagram right here. Notice where the energy state for the reactants is and where the energy state for the products is. Now for any reaction to go to completion, or to convert the reactants to the products, it first needs to achieve the energy of the transition state. Now, the transition state is commonly mistaken for an intermediate. It's not a true intermediate. It's sort of a transient molecule that forms for really split seconds and then rapidly converts into the product. But that means that when two reactants collide with one another, they need to collide with high enough energy to achieve this transition state. And you can see how without an enzyme, that transition state energy is pretty substantial. So that's the activation energy right here without an enzyme. However, once you add an enzyme, you sort of lower the bar. Like the molecules don't have to collide with high enough energy. So notice how, and this is very important and high yield on the MCAT, notice how the enzyme isn't impacting the thermodynamic side of things. It's not like it's increasing the energy of the reactants or the products, and therefore is not impacting change in G at all, because change in G is the difference between the energy state of the reactants and the product. But rather it's just lowering the bar of that transition state or that activation energy. So now let's get to our schematic right here. So here we have molecules, and molecules are constantly in motion. So let's say these are substrate and they're constantly colliding with one another. Now, these green arrows represent the energy with which they're going to collide with one another. So based on our discussion over here, this molecule, when it collides with its neighbor, it's likely not going to result in a reaction because it might go up to here. Similarly, this molecule over here will likely not result in completion of the reaction because it might reach like this point right here. And the thing with transition states or activation energy is that it's an all or nothing process. So you either reach it or you don't. All right, so then we look at this molecule. It looks like it has a lot of energy. So this one will likely result in the achievement of the transition state and therefore will proceed to the product. Now notice what I say over here. A reaction occurs if reactants collide with high enough energy due to chance or by chance. And that's the key. In the body, in reality, it's not like we have 10 or 15 molecules. We have millions, if not billions of molecules. So in order for one with high enough energy to collide with another, it can take 10 minutes, 10 hours, 10 years. So if we were to rely on these sort of reactions for life, none of us will be alive today. Now, what happens when you add an enzyme to the mix? Look, notice that none of these molecules now suddenly have a higher energy. They all collide with the same exact energy, whether the enzyme is there or not. It's just that the enzyme lowers that bar. So now it's not only this molecule that will result in completion of the reaction, but also this one and this one and maybe this one. This one probably has too little energy, so it might not result in a reaction. So not all of them will result in a reaction, but you can see how now we have a bunch of molecules. Once they collide with one another, they will result in the completion of the reaction, and that's all thanks to the enzyme and its stabilization of the transition state and lowering of the activation energy. Now, again, I can't emphasize this enough. Notice how change in G has not changed at all, and again, that's because enzymes do not impact the energy stored in the reactants or the product. All right, the next key properties I wanted to dive deeper in are that enzymes are not permanently altered during a reaction and that they have high specificity. So there have been two models proposed to explain the enzyme substrate binding mechanism. You can see the first one here is the lock and key theory, and this one posits that the enzyme is sort of like the lock, and the substrate is sort of like the key for that specific lock. Like the enzyme, in other words, you can think of it as a mold, and only one structure can fit into there. All right, and then notably, once the reaction goes to completion, you can see that the product, the reactants changed. Now we have a circular shaped product. However, the enzyme snapped back to its original conformation, and that's sort of the idea that enzymes don't get altered permanently after a reaction, and you can reuse the same enzyme for multiple reactions. Now, the other model that's been proposed is the induced fit model. This one posits that enzymes are not necessarily like a mold that fits their substrate, but rather the way they work is that they once they interact, once they encounter their substrate, they will have a conformational shift at the active site to accommodate their substrate. And then once the reaction goes to completion, they'll snap back to their original conformation. All right, our final key property over here, enzyme activity is influenced by environmental conditions such as pH and temperature. So right here, the ideal pH for most enzymes is physiologic pH. Makes sense, you know, our blood is at a pH of 7.4, and that will be the optimal pH for most enzymes in our bodies. However, there are certain exceptions. So for example, in lysosomes, you have enzymes that function optimally at a pH of 5, and this makes sense because our body wants to compartmentalize the function of lysosomes. So within lysosomes, what takes place? Well, it's the degradation and recycling of proteins. And so you only want those enzymes to be able to function inside of the lysosomes so as not to disrupt other key structures within the cell. So if they're in other locations of the cell where the pH is closer to 7.4, those enzymes won't function. And we have similar enzymes with different optimal pHs in the digestive system, for example. Next, let's talk about temperature. So the ideal temperature for many enzymes is around 37 degrees Celsius or 98.6 degrees Fahrenheit, can be a little higher than that. Now, notably, reactions tend to increase. So reaction rates tend to increase as you increase temperature. And this makes sense because as you increase temperature, the molecules start moving faster, the enzymes start moving faster, and therefore reaction rates proceed quicker. However, at a certain point, if you increase the temperature beyond that point, you will have a sudden drop in the activity of the enzyme. And what is that point? Well, let's imagine for a second that we have a test tube and we isolate some enzymes from the human body and we put them at a temperature of 20 degrees Celsius. Well, you'll notice that the enzyme activity might be over here. As you increase the temperature, let's say 30, 35, 37, 40, beyond that point, you increase the temperature further, you have a sudden drop. And that's because what you're doing at that point is you're denaturing the enzymes. So the enzymes are losing their natural three-dimensional shape, they're losing their tertiary structure, and therefore they can't carry their biological activity. All right, cofactors and coenzymes. Many enzymes require cofactors, which are inorganic molecules or metal ions, such as iron and copper, or coenzymes, which are organic molecules, typically vitamins or are derived from vitamins. So as you can see right here, I have vitamin A, E, D, and K in red. These are fat-soluble vitamins, and then B and C are water-soluble vitamins, and that's why I have them in blue. Now, the derivatives of vitamins such as FAD and NAD+ can also function as coenzymes. So holoenzymes, just some terminology over here, holoenzymes are enzymes with their cofactor, and then apoenzymes are enzymes without their cofactor. So obviously without their cofactor, they won't carry their functions to maximal capacity. So that's just a way of regulating enzymatic activity. And then when it comes to prosthetic groups, so those are coenzymes or cofactors that are tightly bound to their enzymes and are necessary for enzymatic activity. All right, if you found this topic a bit difficult to digest, that's normal. This is a difficult topic, and I think enzyme kinetics, the next video, is even more difficult, so be sure to take your time with this topic, and then if you have any questions, be sure to hit me up. You know where to reach me. Don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it.